Welcome to BrainMatters, the show about all things neuroscience. Whether you're a seasoned academic or just curious about the brain, our guests will be covering everything from cutting-edge research to practical tips for improving our mental health. I'm your host, Thomas von Ryan, and today I'm joined by Dr. Philip Ryan, Group Head Researcher at the Florey Institute and Medical Director at a clinical trials centre in Melbourne, here to discuss the role of oxytocin in homeostasis and addiction. Phil, welcome to the show.
Yeah, thanks so much, Thomas. It's a pleasure being here.
So we'll start out with, can you tell us a bit about yourself and the path that your academic career has taken over the years to get where you are now?
Yeah, sure, Thomas. So my background is I was trained as a doctor. So as soon as I left school, I went to do my medical studies at the University of Melbourne. And during my medical studies, I actually took a year off to do what was called a Bachelor of Medical Science, which was a research year. And I did that actually in the field of cancer and haematology. And I really enjoyed that year. Actually, I'd sort of done it as a side year to try something a bit different, and I had a really good time. And had a couple of publications at the end, but I think I just enjoyed the research and the thinking through a lot of the medicine, and how I suppose we come to some of the discoveries that will then impact patients down the track. So after I finished medical school, I did about 3 to 4 years, actually probably more like 5 years, working in different, particularly medical subspecialties, and was considering specialising, but the research bucket hit me. And I decided rather than continuing on with some of the specialties that I'd go and do a PhD in neuroscience. Neuroscience, to my mind, had been particularly fascinating. I'm really interested in behaviour and really interested in, I suppose, the neural circuitry behind it. So I undertook a PhD at the Florey Institute, which is at the University of Melbourne, and looked at a particular peptide that was involved in feeding, drinking, anxiety and depression, as well as having an impact on addiction. So I found it a really interesting topic to sort of get my head around the neural circuitry underlying those different behaviours. While I was doing that, I was keeping up a little bit of my medical side of things too. But at the end of my PhD, I was awarded a fellowship to travel overseas to America. So I actually ended up going to the University of Washington in Seattle. And that was where I discovered this group of neurons that are related to oxytocin, and using a lot of genetic mouse models. So the lab that I went into, the supervisor or the principal investigator was a Howard Hughes medical investigator called Richard Parmiter, and he'd spent many years doing a lot of genetic technologies and say developing transgenic mice. And before I went over there, he developed a transgenic mouse to study the oxytocin system. So it was, a novel system to work with, and it meant that I could study these neurons. And so I managed to discover these neurons, found that they played a role in fluid intake. And then after my 4 years there, came back to Melbourne to help set up a lab and worked a little bit more on this oxytocin system. And while I was back in Melbourne, a few different reasons, but while I continued on with my research, I also decided to look around for something that might use both my medical and research background, and then applied for a role at this clinical trials centre as a principal investigator. So and that's in first in human studies. So I suppose it's the other end of research where drugs, that have been developed in the lab are then finally being introduced into humans. And I found that very interesting as well. So recently been promoted to medical director at the Melbourne site. It's called, the company's called Nucleus Network. And with that, I do quite a few neuroscience trials, but it's not just neuroscience. It's a whole vast range of trials. And while I'm doing that, I've still got a little bit of the research going on at the Florey.
Perfect, thank you. So I really wanted to have you on because when I think when most people think of oxytocin, they think of, love hormone, social connection, but your role and the labs in discovering this cluster of neurons really, it's very different from that conventional understanding. Could you talk a bit more about that discovery and what actually led up to it? What were the stages?
Yeah, absolutely. So you're right. I might even just give a bit of background about oxytocin, because you're exactly right. So I think oxytocin was discovered over 100 years ago, and particularly for its role in childbirth and also in lactation. So both, I suppose for women, that need induction, they have an oxytocin mimetic. often injected into them. And for lactation, oxytocin is heavily involved in that process as well. But as you say, over the last couple of decades, oxytocin's role in social interaction and in that, I suppose, that love hormone, which I suppose is to do with that maternal bonding, has become much more prominent. So the reason why I got into oxytocin was a little bit indirect. So when I was doing my PhD, I was working on a neuropeptide called relaxin-3. I won't go into great details about that, but it does seem to play a role in increasing feeding, decreasing anxiety, and play a role in addiction. And I was interested, after looking at the neural circuitry, to see what was downstream of this. And there seemed to be a role of relaxin-3 in interacting with the oxytocin system. And because the oxytocin system is so fascinating, I thought that I would maybe see if I could do some extra studies on that. So when the principal investigator over in the US, allowed me to join his lab and developed this particular mouse model to study, I thought that would be a really good way of sinking my teeth into oxytocin. Now, my major interest is in the neural circuitry underlying behaviour, so behaviours like addiction or social interaction and anxiety. But mouse models and rat models of these sort of behaviours are quite complex. And I had seen this effect of relaxin-3 on feeding, when I went over to the US, I thought I might start just with some relatively simple behaviours. So I wanted to look at the role of oxytocin in feeding and drinking. And so just a bit of background. So oxytocin, when it's injected peripherally, is known to decrease feeding and actually decrease anxiety and play a role in addiction as well. So it is known to play a role in those, and exactly how that happens is not fully known or understood, but it seems to play a role centrally. So for my first year when I was over in the States, I was looking at some of the downstream pathways. So oxytocin itself is produced in the hypothalamus in two various regions, but it acts on obviously various downstream brain regions all over the brain, actually. And so I looked at some of these downstream brain areas because I was really interested to know that if I inactivated some of these brain regions or activated some of these downstream brain regions that oxytocin acts on, whether or not it might impact feeding behaviour. That was my first initial thought, because I thought feeding behaviour is a really good model to use. And unfortunately, I think in my first year of doing research, I both activated and inactivated about 5 different brain regions in these mice, and found absolutely no effect on feeding, which was quite disappointing because I'd spent a lot of time with these mice.
How are you activating and deactivating these
Yeah.
regions?
No, good question. So with these transgenic mouse models, these have been developed so that the mice are to all intents and purposes like a normal mouse. But when you can inject some viruses that are called DREADs, so designer receptors exclusively activated by designer drugs, and using these transgenic mice, you can add these receptors selectively just to the group of neurons that you want to activate or inactivate. So selectively on the neurons that respond to oxytocin. So I would inject these receptors, say, into a group of neurons in the nucleus accumbens, for example, which is a reward centre and contains a lot of these oxytocin receptors. Wait for about 3 weeks so that the receptor would express. And then you can inject a designer drug peripherally, and it will only activate the oxytocin receptor expressing neurons in the nucleus accumbens, for example. And the effect lasts for about 4 to 6 hours. So it's really a great way of seeing if you activate those neurons and whether or not feeding increases or decreases, for example, or if they have a role in anxiety and depression. I also did some optogenetics, which is very similar as well. I'm not sure if you've heard of optogenetics, but that's introducing a receptor that will respond just to laser light. And so you can inject the same receptors and then apply a laser light through a cannula into the top of the mouse, the mouse brain, and either turn that off and on. And that's actually even, I suppose, more distinct. You can, as soon as you turn the light on, you might see a change in behaviour, and then you turn it off and the behaviour stops. So those are two of the types of techniques that I used. And then you can also do that to inhibit the same groups of neurons as well. One of the other techniques I used, which I could come to as well, was not just activating and inhibiting, but you can actually use what's called calcium imaging techniques to visualize the neurons while the mouse is behaving. And so what will happen is the neurons will fluoresce when calcium is increased. And if you have a miniature microscope implanted, you can watch while the mouse is performing certain behavior, and these neurons will fluoresce while it's performing the behavior, and then they'll turn off afterwards. So there's some really nice fancy genetic techniques that really allow you to selectively stimulate or inhibit or visualize certain groups of neurons. So going on from there, so I tried about 5 different brain areas which had a large amount of oxytocin receptor expression and didn't see an effect on feeding. And the lab that I was in Seattle, a large focus of that lab had actually been on the region of the brain called the parabrachial nucleus, which is not, a really well-known brain region, but it's in the hindbrain, and it seems to have an effect on lots of different behaviours. Including feeding and drinking. And the lab that I was in had really looked at it in terms of its role in feeding behaviour. So there was a group of oxytocin receptor expressing neurons in that particular brain region. And I found when I activated that group of neurons, it actually didn't have an effect on feeding, but it did have an effect on drinking behaviour. So I took water away from the mice overnight, so they should be very thirsty. And then when I activated these neurons and returned water, they just didn't drink much at all over the next couple of hours. And then once the effect of the designer drug wore off, then they'd start to drink again. Whereas feeding, no impact, and it didn't seem to impact too many other behaviours. I've gone on to look at a couple of others, but it seemed to be really quite selective. And then doing some more studies, it seemed to be signalling that that the mouse was satiated for water. So it was quite an interesting discovery. And then from there, I could go on to find out the neural circuitry around that. I also tested, I've said that I'm just stimulating this group of neurons. They do respond to oxytocin, although I don't think oxytocin's the only peptide that acts on these neurons. And interestingly, that group of neurons is just adjacent to the feeding group of neurons, but they're quite distinct. So it's a very small particular brain region, but it looks like it's got very distinct subnuclei that act on very different behaviours.
It's very interesting to hear about the initial 5 clusters you went through first before finally landing on this group. Leading on from that, the Florey Research Group page says that you're looking into oxytocin and this group in relation to the intake of sugary and alcoholic fluids. Where does this work lead, especially with regard to addiction, which is quite common in these fluids?
Yeah. So when I came back to Melbourne, I was really interested in exploring this group of neurons further. So the lab that I'm collaborating with works a lot on addiction, and a lot of their studies are to do with alcohol and sugary drinks. It's also very interesting because a lot of the brain regions that are involved in addiction are also similar brain regions that are involved in, say, the wanting or the liking for palatable food. So there's a lot of interaction. So I wanted to see, I'd seen this effect on fluid intake, particularly water intake, whether it might have an effect on decreasing alcohol, sugary drinks, and other behaviours, other types of solutions. And what I ended up finding was that this group of neurons seems to be quite selective for stopping rehydration. So if I dehydrated an animal overnight and gave it alcohol, it didn't drink for the first bit of time. Or if same with sugary drinks, etc. But if we left it for a period of time afterwards, they still wouldn't continue drinking, say, water, but they would start to drink alcohol or sugary drinks, provided that they were at a palatable level. So what I've discovered, I suppose, is this group of neurons that I've looked at seem to be particularly involved in the homeostasis, the fluid homeostasis pathway, so the rehydration, dehydration. So if you're drinking alcohol just to rehydrate or drinking sugary drinks to rehydrate after you've been dehydrated, it'll stop that. But it won't stop drinking for the liking sensation. So they won't drink a whole lot in a short space of time. They'll just drink it because they like it over a period of time. So that was quite interesting. And then subsequently, as well as looking at that, as I said, in this parabrachial nucleus, there does seem to be groups of neurons that are surrounding that, that seem to be involved in different aspects of fluid behaviour and feeding behaviour. There does seem to be another group of neurons, which I'm quite interested in, which do seem to play more of a role in palatable drinking and may be involved in, say, the decreasing of just the palatable intake, rather than just the homeostatic fluid intake. That's sort of, more preliminary, but I'm really quite excited to look at some of these different brain regions.
Right, because my brain would immediately jump to dopamine with regard to addiction and asking whether there's would be any interaction there. But it doesn't seem to be the case if they're so separate from homeostasis versus addictive behaviors.
Yes, so it's a really good question. So I think for the group of neurons that I've looked at, there doesn't seem to be too much of an interaction with dopamine. But being in an addiction lab, I'm always very interested in dopamine. We talk about dopamine all the time. So I'm actually interested. So, firstly, in terms of just going back a step, does oxytocin and dopamine interact? So, I think not with this group of neurons, but there does seem to be definitely an interaction between dopamine and oxytocin. Um, just looking through the literature and my understanding is I think oxytocin might direct some of the sort of the salience behaviour into more the social and make it maybe interact in that perspective. But the other really interesting thing is that oxytocin does seem to play a role in decreasing addictive-like behaviours. Now, exactly the way that occurs, I'm not sure, but presumably there is some interaction with the dopamine system for that. I suspect that's not through this group of neurons in the parabrachial nucleus. I've been trying to do a study on that in the last year, and there didn't seem to be as much of a role in that. I suspect it's other parts of the brain. Probably, regions such as the nucleus accumbens or some other brain regions which also express quite a lot of the oxytocin but play a role in dopamine, have a lot of dopamine signaling as well. But that's something that I'm very interested in. And not just in terms of my research, but also in terms of the clinical trials. We seem to be testing some, there have been sort of some, not oxytocin related, but similar sorts of compounds that some companies are starting to develop that might, that are oxytocin like, that might play a role in the addictive type behavior. So I find that area really fascinating.
Given all of your background and the research that you've done, has this work changed any habits you have in your daily life or any recommendations that you give friends now?
Yes. So the short answer to that is probably not. But I just want to go back a couple of steps. I suppose the whole reason why I got into this research and that I find it fascinating is I am, I find human behavior really fascinating. And I think traditionally, really, the brain circuits and the underlying brain mechanisms have been a bit of a black box up until probably the last decade or two, where we've had some much better tools to tease them apart. I mean, traditionally, we've been able to sort of inactivate a particular brain region more by cutting it, whereas now with some of these selective techniques, we can really activate or inhibit it very specifically. And I suppose it's really showing that very small groups of neurons impact on various behaviors. And I think teasing that apart is really interesting. But then thinking about it, so behaviors like addiction and anxiety, which often have quite a lot of stigma in society, particularly addiction, I think actually understanding the neural circuitry that forms a basis in the changes that can occur with addiction has actually probably made me a bit more both self-aware but also a bit more compassionate towards people. I mean, I think a lot of these drugs really change a lot of they cause neuroplasticity. They make big changes in the brain, which are very difficult to control. I mean, I think sometimes there's a bit of a push, say, amongst the media or certain people, that it's all about self-control. And perhaps there's an aspect, but there's definitely some brain changes. So if there's a way we can understand these brain changes, be able to selectively target them, and then perhaps find some treatments for them, I think it actually makes people, and it's made me and other people, I think, around it a bit more compassionate, because you can understand and then perhaps treat. So I think neuroscience really can impact, I suppose, how we think about conditions and also how we potentially treat them as well, and make us a bit more compassionate.
I think that's a very positive outlook, definitely. And finally, I touched on it briefly in your introduction, but you're now working as a medical director, a principal investigator in a bunch of clinical trials. Can you tell us a bit about the trials that you're working on at the moment, what we've got to look forward to in the future?
Yeah, sure. So, I mean, obviously, I can't go into great detail because there's confidentiality, but I can paint a bit of a broad brush. So I'm particularly involved in phase one clinical trials. So they're the trials where they're first introduced into healthy volunteers. A large factor in these trials is looking at safety and tolerability. So we're very careful with starting at very low doses and then gradually building up, usually single doses first off, and then maybe some multiple doses. Um, and pay particular caution, to make sure that we understand that there's no, major adverse events from these drugs or anything like that. And then we check the pharmacokinetics or how the drug interacts with the body. But some of the indications that I've been particularly interested in are to do with some of these. There have been some, say, Ozempic-like type drugs, so those sorts of things. So they're quite interesting. Some neurological type drugs for Alzheimer's disease, for indications such as cystic fibrosis. There's a broad range of cardiological, asthma-related drugs, and there's also quite a lot of autoimmune-related drugs, things like for conditions like ulcerative colitis, which affects the bowel, and various other conditions. We also look at, I suppose, some. It depends on the drugs, but we also do look at some drugs which might impact on some cancer treatments as well.
Amazing. And all the best of luck with it.
Yeah, thank you.
Now, before we wrap this up, I would like to ask some light-hearted questions that should give people a bit more of a glimpse at the person behind the scientist. Does that sound good?
Sure.
It'll be simple. You're in Australia, so I have to ask you, what is the go-to coffee order in all your lovely cafes?
I really tend to go for a cappuccino, but the flat whites are also very good as well. And there's some really nice coffee places, both close to my home and also close to Melbourne University. Seven Seeds is good. There's some really good ones. I'm not sure if you checked out a few around Melbourne University, but there's some great coffee. And actually, we're very close at Melbourne University to Lygon Street, where there's some really nice cafes as well.
Now, we've just talked a bit about addiction behavior, fluid intake, and food. It's been a long day at the office. What's the go-to takeaway order, Phil?
Well, the takeaway order is generally, because I've got teenage kids. It's often generally a sundae or a McFlurry at McDonald's. That's typically for the kids, but because I'm ordering three, there's usually an extra one that's added to the side.
Um, Australia did very well in the Olympics this year. If
And
you had 1 year to get trained up and take part in a sport, what would be your event of choice?
Oh, I'm a runner, Thomas. So I at school I used to love and got involved in the 800 meters and the 1500 meters, so the track events, and used to get involved in some of that. And so more recently, because my kids are getting much better than I am, they're teenagers now. We've been doing quite a few of the 5 and 10K runs around Melbourne because there's lots of fun runs, particularly at this time of year. Now, I haven't done this yet, but I would really be keen to do, half marathon first off, but a marathon. I reckon that would be great. It would take a lot of work. I'd love to do the training, but that would be my key. The only thing is, I know as an Australian, it's probably unlikely that that I'd win a gold medal in that event, but I do really enjoy it. That tends to go to some of the other nations, for sure.
Finally, are there any, apart from running, are there any hobbies that you like to spend your free time on the weekends away from research?
Yeah, sure. So, in time gone by, it was probably a bit of music. I played a bit of piano. Can I be honest now? I've got three teenage kids. So my weekend is definitely taken up by sports events. A lot of they're actually, all three are very much involved in some of the school musicals as well. And I find that really takes a large proportion of my time on the weekend. But I do enjoy watching, say, the footy, so Australian rules football on the weekend.
Okay, that's perfect. We've covered a lot, and I think you've given people a really good introduction to your work on this new dimension of oxytocin. So while you have this platform, is there anything that you or your lab is doing that you would like to promote?
Yeah, thanks, Thomas. So I mean, there's a couple of things that I'm currently working on with the lab. So one is expanding on this role of oxytocin, seeing its role in, say, anxiety and addiction-like behaviours, and I think there's some really interesting projects there. I'm also doing and planning to do. I've done some preliminary studies on salt and stress intake. A lot of the fluid-related areas are also interact with salt because salt and fluid are, combined a lot. And I think that the role of salt in stress is really interesting. So I'm starting to do a bit of research in that line of things, looking at a couple of different brain areas, but they're very sort of similar to the sort of brain areas that I've been looking at with the oxytocin system. And I suppose the other area that I'm also looking at is going back a little bit to my PhD. I looked at a particular peptide called relaxin-3, and I'm actually starting some collaborations to look a little bit further into that peptide, which seems to play a role in feeding anxiety and addiction. And I'm really interested in exploring that a bit further with some collaborators.
And if people wanted to reach out to you for collaboration or discussion on these topics, where could they do so?
Yeah, sure. So I think probably the best way of looking is at the Florey website. And you can look up my page, which will be under Philip Ryan, and there's some contact details there. So I'd be very happy to chat if anybody's very interested in this sort of work.
All right, Phil, that concludes our conversation. Thank you so much for your time today.
Thanks so much, Thomas. Really appreciate it.
If you enjoyed this discussion and want to learn more about the brain, why not check out our website at brainmatters.nl, where you can find easy-to-follow articles in both English and Dutch to satisfy your curiosity. I would also like to thank the State Library of Victoria for providing us with the space to record the episode. Until next time.